Welcome to the Learning Module for Recombinant DNA and Biotechnology in Medicine. This is Dr. Tafke for the Cellular and Molecular Medicines course. After completing this learning module, students should be able to describe the common tools and techniques used in molecular cloning, apply knowledge of southern blotting, northern blotting, and western blotting, explain the steps in molecular cloning of a gene, apply knowledge of restriction fragment length polymorphisms and their usage, apply knowledge of allele-specific oligonucleotides in dot blot analysis and PCR, explain the concept of DNA microarrays, explain recombinant proteins, list their advantages over biological sources, and provide examples of their usage. Describe gene silencing with double-strand RNA through the RISC complex, and describe the different types of gene therapies and give examples. In Part 1 of this learning module, we will have an overview of the tools and techniques used in recombinant DNA technology. This will include restriction enzymes, gel electrophoresis, DNA blotting and hybridization techniques, including southern blotting, northern blotting, and western blotting, reverse transcription, polymerase chain reaction or PCR, and molecular cloning techniques.
Are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering. At high temperatures, double-stranded DNA will denature or separate into single strands. When the temperature is lowered, the two strands will anneal because of the base pairing interactions of the complementary strands. If the nucleotide sequences are similar, complementary strands from different sources will also anneal. This is called hybridization, to indicate that each strand of DNA came from a different source. Regions of double-stranded DNA in which the respective single strands hybridize to one another are said to be homologous, or have similar or identical nucleotide sequences. The high specificity of base pairing interactions between complementary strands of DNA can be used to locate a specific nucleotide sequence in a sample. If the DNA from one source is immobilized by attachment to a solid surface such as nitrocellulose, homologous DNA from another source will hybridize and be retained by the immobilized DNA. This is the basis for various DNA probe techniques. Non-homologous DNA will not attach.
These tubes contain identical DNA fragments, but they will be cleaved with different restriction enzymes to yield fragments of different sizes. Enzyme 1 cuts the DNA into fragments A and B, which we color for tracking purposes. Enzyme 2 cleaves the DNA at a different recognition sequence, yielding fragments C and D. Adding both enzymes yields fragments A, E, and D. Gel electrophoresis is one of the most useful means of separating and purifying DNA fragments for further analysis. In this technique, a jello-like slab of material called agarose is molded with wells, placed in a buffer solution, and hooked up to positive and negative electrodes. The DNA solutions, to which blue dye is added, are then pipetted into the wells. A well is also reserved for the placement of DNA of known sizes, and then the power supply is turned on. The blue tracking dye is negatively charged and migrates toward the positive electrode, as does the DNA. The DNA backbones contain negatively charged phosphate groups, which are attracted to the positive electrode. The smallest fragments move the fastest, being entangled less in the agarose matrix of the gel. When the blue dye reaches the bottom of the gel, the power is turned off. A fluorescent dye is then used to stain the DNA fragments. A DNA fragment in the size standards lane is the smallest, in this case about 500 base pairs, followed by DNA fragment D. The largest fragment is 23,000 base pairs long. Each band of DNA may contain millions of fragments of the same size. In many cases, a researcher may want to determine which DNA fragment contains a DNA sequence of interest. To do this, the researcher prepares the DNA in the gel to make a copy, known as a blot. First, the gel is soaked in a basic solution so that the double-stranded DNA denatures into single strands. The gel is then transferred to a salt solution and a nylon filter is placed on top of the gel. Absorbent towels are placed on top of the filter. The salt solution draws the DNA through the gel toward the nylon filter, where the DNA adheres. The filter is treated so that the DNA adheres to it permanently, and then the filter is placed in a solution with a radioactive probe. The probe consists of single-stranded DNA that is complementary and will hybridize to the band of interest. The filter is washed to remove any unhybridized probe. A piece of X-ray film is placed over the filter. The radioactive probe exposes the film, revealing the locations of hybridization. Knowing which bands contain the sequence of interest, an identical agarose gel can be created, and a band of interest can be cut out of the gel. The DNA in this band can then be manipulated for further analysis.
The southern blot is used to verify the presence or absence of a specific nucleotide sequence in the DNA from different sources, and to identify the size of the restriction fragment that contains the sequence. In this procedure, the DNA is isolated from each source and then digested with a specific restriction enzyme. The DNA restriction fragments are then loaded onto an agarose gel and the fragments separated by electrophoresis according to size, with the smaller fragments migrating faster than larger fragments. The fragments are made visible with ethidium bromide. The DNA is then transferred from the fragile gel to a nylon filter. Next, the radioactively labeled nucleic acid probe is added. The probe binds to complementary DNA segments. Note that the DNA segment being probed is not present in organism B. To detect the position of the radioactive probe, the nylon membrane is covered with an X-ray film. After development, the positions of the probe become visible. In most eukaryotes, the expressed segments of the gene, called exons, are separated by intervening sequences of nucleotides, called introns. The exons and introns are transcribed by RNA polymerase, generating what is called precursor messenger RNA. The introns are then excised from the precursor messenger RNA, and the exons linked together to form the mature messenger RNA. Prokaryotic cell DNA does not contain introns, and prokaryotic cells are therefore unable to remove the introns from eukaryotic DNA and make functional messenger RNA. Therefore, eukaryotic DNA cannot be cloned directly into prokaryotic cells to make useful eukaryotic proteins. Before eukaryotic DNA can be cloned into prokaryotic cells, the mature eukaryotic messenger RNA must be isolated and used to make DNA without introns. The enzyme reverse transcriptase is used to convert the purified eukaryotic messenger RNA into double-stranded DNA. The resulting DNA, called complementary DNA or cDNA, can then be cloned into a bacterial cell. Genes cloned as cDNA can be transcribed and translated by the bacterial cell machinery. The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
There are two primary mechanisms for utilizing PCR to obtain a clone from eukaryotic cells. You can utilize PCR to directly isolate copies of genomic DNA, which then contain all regulatory regions as well as introns in the region of interest. The other method couples reverse transcription with PCR and is called RT-PCR, in which messenger RNA is first isolated from the cells and then subjected to reverse transcription to produce a cDNA copy of the messenger RNAs. The cDNA copy then can be amplified using PCR. The resultant PCR product will lack introns and as a result can be expressed in a prokaryotic organism. Cloning vectors are vehicles in which DNA from one organism can be introduced or inserted into a host cell of another organism. There are several varieties of them, including plasmids, bacteriophages, bacterial and yeast artificial chromosomes. The major difference between all of these being the size of DNA that can be inserted into that vector. Plasmid vectors are double-stranded circular DNA molecules capable of autonomously replicating in a host bacterial cell. Here's a diagram of a prototypical plasmid vector known as PUC18. Plasmid vectors will have an origin of replication, which allows it to be autonomously replicated in the host bacterial cell. It will have a polylinker sequence to facilitate cloning with a variety of restriction enzymes. Plasmid vectors will also carry sequences for antibiotic resistance and to aid in selection of bacteria that have taken up the plasmid. The following animation illustrates the basic functions of plasmids in molecular cloning.
Plasmids can be good cloning vectors because they carry an origin of replication and are therefore able to replicate independently within a cell. Most plasmids used as vectors also encode some type of selectable marker, such as the gene for resistance to ampicillin. If the host cells are ampicillin sensitive, the only host cells that can grow on a medium containing ampicillin are those that have taken up the plasmid. Vectors must also have a small sequence of base pairs that can be recognized by a restriction enzyme. When this enzyme opens the circular plasmid, foreign DNA can be incorporated. When the plasmid vector and foreign DNA are both cut with the same restriction enzyme and mixed together, not all molecules will join to form recombinants. Some vector molecules will reanneal without incorporating foreign DNA. To identify cells that contain plasmids that have incorporated foreign DNA, a second marker gene is needed on the vector. This second marker contains the restriction enzyme site within its nucleotide sequence. If foreign DNA is inserted, the second marker is inactivated. This is referred to as insertional inactivation. A common second marker is the LAC-Z gene, which codes for the enzyme beta-galactosidase. Beta-galactosidase can cleave a colorless chemical called XGAL to form a blue compound. Therefore, colonies of cells that harbor the intact vector but no new recombinant DNA can make beta-galactosidase and form a blue color in the presence of XGAL. However, colonies that contain new recombinant DNA cannot make beta-galactosidase and are white. The first step in cloning a gene is to isolate the DNA from the organism that contains the desired gene. The isolated DNA is purified and then fragmented with a restriction enzyme. Restriction enzymes used in cloning produce staggered cuts in specific sequences in the DNA, generating fragments with cohesive ends. Each fragment has a single-stranded sequence of nucleotides on its ends that is capable of hybridizing with DNA that has been fragmented with the same restriction enzyme. The DNA fragments are then incorporated into plasmids. The type of plasmid used for cloning has a single restriction site, and when cleaved by the restriction enzyme, generates the same cohesive ends that are in the fragments of the DNA to be cloned. The cohesive ends of the plasmid and DNA fragments now line up, and the enzyme, DNA ligase, is used to form phosphodiester bonds. The next step is to incorporate the plasmids into bacterial host cells by transformation. Each cell contains a different segment of DNA from the original organism. Taken together, these cells represent a DNA library. The cells can now be plated out on an agar medium. The colony of cells containing the desired cloned gene can then be identified and isolated.
Recombinant DNA technology has revolutionized the diagnosis of disease. In this next section, we will examine how restriction fragment length polymorphisms are used to analyze genetic disorders, how allele-specific oligonucleotides can be used as probes in hybridizations or as primers for PCR to detect mutations, and how microarray technology now allows for genotyping or single nucleotide polymorphous analysis, as well as to study gene expression. One of the first methods for analyzing genomic DNA for disease-causing mutations were variations of the southern blotting technology, as illustrated in the following animation. RFLP analysis is also used, or was, for paternity issues and in criminal investigations.
Restriction enzymes recognize very specific sequences of nucleotides in DNA. DNAs from different individuals rarely have exactly the same array of restriction sites and distances between these sites. Therefore, the population is said to be polymorphic, having many forms for these restriction fragment patterns. These differences are referred to as restriction fragment length polymorphisms, RFLPs. Such differences may arise through mutations. By cutting a DNA sample with a particular restriction enzyme, DNA fragments of different length are obtained. These fragments are separated by gel electrophoresis. This provides a pattern of bands that is unique for the particular DNA being analyzed. These DNA fingerprints are used in forensic science during criminal investigations. RFLPs are also useful as markers to identify particular groups of people at risk for certain genetic disorders.
This slide illustrates how RFLPs can be used to analyze the DNA of family members for the presence of a disease-causing allele. In this hypothetical example, allele A has 3 BAMH1 sites, BAMH1 being a restriction endonuclease, while the mutant allele, allele lowercase a, has only 2 BAMH1 sites. Thus, digestion of the wild-type allele will produce a shorter DNA fragment than restriction enzyme digestion of the mutant allele. After southern blotting of the restriction enzyme digested DNA with the probe that is common to both the long and short BAMH1 fragments, it is possible to genotype a family. Persons homozygous for the wild-type allele will only produce the small fragment, while persons homozygous for the mutant allele will only produce the large fragment. Heterozygous individuals will produce both a small and large fragment. In this example, we can see that both parents were carriers of the mutant allele. They had one daughter who was homozygous for both mutant alleles. They had one son who was homozygous for the wild type, and one heterozygous son. DNA fingerprinting is a form of restriction fragment length polymorphisms. It is used to identify individuals based upon variable number tandem repeats in their genomes. Our genome contains numerous segments of repetitive DNA located at specific regions. The number of these repeats is a heritable trait. Digesting DNA with a restriction enzyme that cleaves on each side of one of these locations then will produce fragments of differing lengths depending upon the number of repeat sequences present in that DNA. Digestion of one person's DNA and analysis of the VNTRs at several locations will provide a unique marker for that individual's DNA, or a DNA fingerprint.
DNA fingerprinting is a method of identification based on an individual's DNA. In traditional DNA fingerprinting, the DNA is collected from cells such as a blood sample and cut into small pieces using a restriction enzyme. This generates thousands of DNA fragments of differing sizes. The fragments are then separated on the basis of size using gel electrophoresis. The separated fragments are then transferred to a nitrocellulose or nylon filter. This is called a gel blot. The DNA fragments within the blot are permanently fixed to the filter, and the DNA strands are denatured. Radio-labeled probe molecules are then added that are complementary to sequences in the genome that contain repeat sequences. These repeat sequences tend to vary in their length among different individuals and are called variable number of tandem repeat sequences, or VNTRs. The probe molecules hybridize to DNA fragments containing the repeat sequence, and excess probe molecules are washed away. The blot is then exposed to an X-ray film. Fragments of DNA that have bound the probe appear as dark bands on the film. As seen here, the samples designated 1 and 2 have a different pattern of bands, indicating that they came from 2 different individuals. Traditional DNA fingerprinting has been superseded by an automated procedure in which microsatellites, which also contain variable repeat sequences, are amplified by PCR. The small amplified DNA fragments are fluorescently labeled and subjected to high-resolution gel electrophoresis. A fluorescence detector determines the amount of DNA in each amplified fragment, thereby generating a graph that depicts the pattern of fragments in a particular individual.
Allele-specific oligonucleotide probes are short pieces of synthetic DNA that can be complementary to the sequence of a target DNA. Allele-specific oligonucleotides can be used as probes in dot blot analysis of genomic DNA. Dot blots are similar in methodology to southern blots, except the genomic DNA is not digested with restriction enzymes and separated by size by gel electrophoresis. Instead, the genomic DNA is spotted onto a membrane and immobilized in that location. A labeled probe then is hybridized with the membrane, and if the probe hybridizes to any complementary sequences in the genome, it will be visualized as a spot on the membrane. This slide illustrates how dot blotting with allele-specific oligonucleotides can be used to determine if individuals are carrying the mutation that causes sickle cell. Genomic DNA is isolated from 6 individuals and blotted onto duplicate membranes. One of the membranes is hybridized to a probe complementary to the wild-type sequence, and the other membrane is hybridized to a probe complementary to the sickle cell mutation. There is only a single nucleotide difference between the 2 probes. However, the hybridization conditions are manipulated such that only 100% base pairing between the probe and its complementary sequence is enough for the probe to stay on the membrane through all the washing steps. Genotyping the individuals, then, is just a matter of determining which probe annealed to DNA from which individual. For example, individual 1 was detected by the wild-type probe, but not by the mutant probe, so this individual is homozygous wild-type, as is individual number 4. On the other hand, DNA from individual number 2 was only detected with the S probe and not by the wild-type probe, so this individual must be homozygous for the sickle cell mutation. DNA isolated from individuals number 3 and 5 was detected when probed with either the A or S probe, so these individuals are heterozygous for the mutation and are carriers. Allele-specific PCR methods take advantage of the fact that primer extension requires an absolute match at the 3' end of the primer in order to produce a product. Utilizing carefully designed primers carrying exact 3' matches and 3' mismatches makes it possible to determine if a person is carrying a specific point mutation associated with a disease. This example illustrates how allele-specific PCR can be used to genotype for the presence and absence of 2 alleles differing by a single base. This requires 2 parallel PCR reactions. The first reaction uses a primer designated P1, which is 100% match for allele 1. However, it has a mismatch at the 3' end for the variant allele 2. The reaction is run in the presence of P3, a common downstream primer with 100% match for allele 1 and allele 2. so PCR reactions run with primers P1 and P3 will yield a product if allele 1 is present in the genomic DNA, as is the case in persons homozygous for allele 1 or heterozygous for allele 1 and allele 2. Conversely, the absence of a PCR product means that allele 1 is not present in that genomic DNA and suggests that the person is homozygous for allele 2. If, instead of using P1 in the PCR reaction, you use a second primer designated here as P2, which is a 100% match for allele 2 but has a 3' mismatch for allele 1, you should get the opposite result. A PCR product from allele 2 but no product from allele 1. In this example, individuals A and C are heterozygous, producing PCR products from both sets of primers, while DNA isolated from individual B only produced a product with the P2-P3 PCR reaction setup. Therefore, this individual must be homozygous for allele 2. PCR amplification of genomic DNA can also identify deletions that cause disorders and diseases. The most common mutation leading to cystic fibrosis is a 3 base pair deletion known as the delta F508 mutation. PCR amplification of genomic DNA using primers that flank the position 508 in the CFTR gene will produce a PCR product that is 3 base pairs smaller than the wild-type product.
A microarray, or DNA chip, is a spatial array of oligonucleotide probes, closely arranged on a small solid support surface. The oligonucleotide probes, representing nucleotide sequences in known genes, are synthesized on the solid surface in such a way that both the position and nucleotide sequence of each probe are known. Microarrays can be used to identify organisms and specific genes by hybridization of DNA from the organism to the oligonucleotide probes in the microarray. There are several ways to make a microarray. The one described here involves a methodology adapted from computer chip technology. A piece of glass, about 1.5 by 1.5 centimeters, is covered with a mask containing holes corresponding to the sites of attachment of the desired nucleotides. The first nucleotide to be attached is added and made to bind by a light activation reaction. The mask is then replaced by another mask which exposes sites for the addition of the next nucleotide. This process is repeated until all of the oligonucleotide probes, each with a known sequence and position, is synthesized. Although only 5 nucleotides are shown in the oligonucleotides of this animation, there are usually 15 to 25 nucleotides in length in the microarray. The DNA from the organism being studied is isolated, fragmented, and tagged with a fluorescent dye. The DNA fragments are then incubated with the chip. DNA from the organisms that is complementary to the DNA in the microarray bind together, and the unbound DNA is washed away. The surface of the microarray is then scanned with a laser beam, and the data obtained is used to produce a visible image. Color intensity indicates the extent of hybridization to the different probes. Over 200,000 probes can be constructed on a single microarray. With this technology, any desired sequence of nucleotides can be constructed to form a probe. The presence of genes for antibiotic resistance and other traits can easily be determined, and gene expression can be monitored by hybridizing cDNA obtained from messenger RNA.
Essentially, DNA microarrays are hundreds of thousands of dot blots run in reverse at the same time. The amount of information obtained can be enormous. It is this aspect of DNA microarray technology that is driving the field of bioinformatics and leading us into the new field of personalized genomic medicine. The third part of this learning module will be an overview of the advancements made in the treatment and prevention of diseases made possible by recombinant DNA technology, including the production of therapeutic recombinant proteins, vaccines, and gene therapy. By now, everyone should recognize the most famous protein in the world, insulin. Insulin was the first gene to be sequenced, it was the first protein to be sequenced, and it was the first recombinant protein to be approved by the FDA for use in humans. As I'm sure you remember from Dr. Robinson's lectures, insulin is produced as a pre-hormone in which an A chain and B chain are separated by a sequence that is post-translationally removed by proteases. The A chain and the B chain are then linked through disulfide bonds. Since prokaryotic organisms do not express the proteases necessary to process proinsulin to its active form, the coding sequences for the A and B chain were cloned separately. A vector containing the A chain and a vector containing the B chain of insulin were then transformed and expressed in E. coli. The A chain and B chain were then purified from the E. coli and combined together under the appropriate conditions to allow refolding and formation of the disulfide bonds of active insulin. Since then, variations of the same recombinant DNA technology used to produce insulin has resulted in the production of hundreds of recombinant therapeutic proteins for use in medicine. Use of recombinant technology for therapeutic proteins over isolation from biological sources has several advantages, including high purity and specific activity, a steady supply, batch-to-batch consistency, and a much lower risk of immunoreactivity in the patient. Small proteins, such as insulin, or proteins that retain activity in the absence of significant post-translational modification, can be expressed in simple prokaryotic or eukaryotic organisms like E. coli and yeast. However, proteins requiring post-translational modifications for activity have to be expressed in more complex, higher organisms to produce a biologically active product. Therefore, insect cells or cultured mammalian or human cells are used for the production of some recombinant therapeutic proteins. The complexity of the protein and post-translational modifications required for activity are one of the reasons these therapies are so expensive. A vaccine is a harmless variant or derivative of a pathogen that stimulates our immune system. Traditionally, vaccines were either particles of virulent viruses that had been inactivated by chemical or physical means, or active virus particles of a non-pathogenic strain. Recombinant DNA technology is now utilized to produce vaccines. In the cases we've looked at so far, recombinant proteins have been used to supplement a protein which is absent or in low supply. However, there are numerous conditions in which suppressing the activity of a gene may be beneficial. Gene silencing is a relatively new recombinant DNA technology that may allow this to become a reality. Introduction into cells of chemically synthesized double-stranded RNA complementary to a particular messenger RNA sequence will activate the host cell's RNA-induced silencing complex mechanism and thus inhibit the production of the protein. The double-stranded RNA is processed by dicer into small interfering RNA that can associate with the RISC complex, which uses the small interfering RNA to target the messenger RNA that has complementary sequences to it for degradation. There are several RNA interference therapies currently in clinical trials, including one for neovascular age-related macular degeneration. in which double-strand RNA targeting the vascular endothelial growth factor, or VEGF, which is overexpressed in this condition, are introduced directly into the eye.
RNA interference is a mechanism of silencing gene expression after the gene has been transcribed. This interference is due to the presence of antisense RNA, which is complementary to a specific cellular mRNA. The complementarity between the antisense RNA and the cellular mRNA causes the two molecules to bind to each other. The antisense RNA can come from three sources. First, it can be introduced into cells by researchers. Second, certain viruses may produce antisense RNA as part of their life cycle. And third, the integration of genes into chromosomes next to endogenous gene promoters can sometimes cause the synthesis of the antisense strand of RNA. The current model of RNA interference involves two steps: creation of an RNA-induced silencing complex, RISC, and then the degradation of cellular mRNA by the RISC. Creation of the RISC occurs when double-stranded RNA is found in the cell. This double-stranded RNA is recognized by an endonuclease known as dicer that cuts it into pieces that are 21 to 23 nucleotides long. An antisense RNA fragment can then associate with proteins to form the RISC. The silencing complex can then associate with cellular messenger RNA that has a region of sequence homology with the RNA fragment in the RISC. The messenger RNA is then degraded. The presence of this rapid degradation mechanism can reduce and even effectively eliminate translation of the messenger RNA for the gene in question, thus interfering with gene expression.
Gene therapy involves the insertion of normal genes into human cells in order to correct a genetic disorder, in most cases a recessive disorder. There are two types of gene therapy: germline gene therapy, which is transfer of DNA into the germline, which makes the modification inheritable. This is routinely done to produce transgenic animals. However, in humans, there are moral and ethical as well as legal issues that prevent this type of therapy from going forward. And somatic cell gene therapy, which transfers DNA into somatic cells, which is not passed on to progeny. It usually is targeted to a specific organ or tissue, such as muscle or liver. There are two varieties of somatic gene therapy: ex vivo, in which an individual cells are sampled, normal copies of the defective gene are introduced into those cells, the modified cells are then returned to the donor, and if all goes well, the cells produce a normal gene product and the disease symptoms decrease or disappear. The other form of somatic gene therapy is in vivo somatic gene therapy, in which therapeutic genes are introduced directly into cells in the body. There are several ways of introducing genes into cells for gene therapy, including RNA viruses or retroviruses and DNA viruses like the Adenovirus family, as well as non-viral methods such as encapsulating in liposomes or direct DNA injections. Mostly for reasons of high efficiency, viral vectors are the preferred method for gene therapies. Viruses replicate by inserting their genome into a host cell. RNA viruses, or retroviruses, integrate their genome into the host genome. This is stable insertion of the transgene that will be passed on to daughter cells if the cells divide. DNA viruses, such as the adenoviruses, do not integrate into the host cell genome. This means that the expression of their transgene is transient and will not be passed on to daughter cells. This means that gene therapy with DNA viruses may have to be repeated occasionally to continue to control the condition. The genomes of RNA viruses are double-stranded RNA. When they infect host cells, the RNA is converted into DNA, which is then integrated into the host cell genome. The therapeutic gene then that is packaged in the retroviral vector can be transcribed to messenger RNA and translated into a therapeutic protein to produce an effect. There are several advantages and some disadvantages to utilizing retroviral vectors for gene therapy. The genomes of retroviruses tend to be very small and limited in the ability to hold exogenous DNA, usually less than 8,000 base pairs. Retroviruses have a limited cell target. They require an actively dividing cell for infection and specific cell receptors for the infection. They are highly efficient, usually one virus particle per cell. In addition, the infection is stable due to integration into the host cell genome. All daughter cells will inherit the transgene. However, since the insertion for most retroviruses are random, there is the potential for insertional mutagenesis, which occurs when insertion of the retroviral genome into the host cell genome disrupts a gene involved in control of cell proliferation, resulting in a hyperproliferative cell. Adenovirus vectors do not integrate their genome into the host cell genome. Instead, the host cell machinery transcribes and then translates the viral genome into protein products which produce the effect in the cell. There are several advantages and disadvantages to using adenoviruses as vectors for gene therapy. The inserted gene can be much larger than that packaged into retroviral vectors. It allows transduction of non-dividing cells and can affect a broader range of cell types. However, the infection is not stable. Therefore, after a period of time, the protein production drops off, so it may require subsequent reinfections with the adenoviral vector. A major limitation in retreatment is that these vectors tend to be immunogenic, which can reduce the effectiveness of retreatments, or worse, cause a hypersensitive reaction and death. Adenosine deaminase severe combined immunodeficiency was the first genetic disease cured by gene therapy. Remember from purine nucleotide lectures that deficiency of adenosine deaminase leads to an abnormal accumulation of purine metabolites, most notably deoxyadenosine, that is toxic to T and B lymphocytes. Therefore, these individuals do not develop a functional immune system. So they, like the boy in this picture, have to survive in a sterile environment. The initial gene therapies developed in the 1990s for ADA-SCID involve transferring the ADA gene into a genetically disabled retrovirus and then infecting T cells that have been isolated from the SCID patient. These cells were then cultured and expanded ex vivo until they had enough cells to transplant back into the SCID patient. These cells grafted back in with the other cells and began to express the recombinant adenosine deaminase, which then allowed the T cells to survive and secrete factors that improved the survival of the B cells, and these individuals that received the therapy developed functional immune systems. This therapy had to be repeated periodically to replace the T cells because T cells were mature cells that had terminally differentiated and thus had a limited lifespan. Subsequent therapies infected stem cells, which then were stimulated to develop into T cells and B cells, making the transgene much more stable in the individual and not requiring retherapies. Mutation in the SCID-X1 gene, which encodes subunits of the gamma C chain found in cytokine receptors found on many immune cells and immune cell precursors, results in defective development of functional immune system because T cells and natural killer cells cannot respond to cytokine signals necessary for their maturation. As with the gene therapy used to cure ADA-SCIDs, stem cells from bone marrow of ex-SCID patients were isolated and infected with the retrovirus carrying the gamma C gene. The transduced cells were then reintroduced into the patient. In most cases, the therapy worked very well, and patients developed a functional immune system. However, in a few cases, the patients developed a leukemia-like condition with clonal T-cell proliferation due to a random insertion of the retroviral vector in or near a proto-oncogene called LOM2. This is an example of insertional mutagenesis. Here is a short quiz to test how well you've mastered recombinant DNA biotechnology in medicine.